So this yesterday we were discussing about the bioreactors, and what I told you that bioreactors are large vessels in which in the nutrient medium the inoculum is put. And what is inoculum? Inoculum is a small quantity of material which have our transformants. Transformants means those bacteria in which our recombinant DNA is there. So that is put, and what happens that after when it is stirred properly, stirred, and when this oxygen mixes it in, and they get sufficient nutrient for their growth, they flourish. They grow in large quantity, and the biomass of them increases. So yesterday I told you about the simple stirred type, and the second one was sparge stirred type. I told you then that in the sparge stirred type of this vessel bioreactor, we have a rectangular tank, and in the center of the tank we have a air which enters in is under high pressure, so bubbles are made, and these bubbles they enter the water, and many bubbles are created in water, and the inoculum which is there in the nutrient solution gets sufficient nutrient which is in the form of vitamins, salts, minerals, plus oxygen, large quantity of oxygen, and they flourish, they start growing, and very soon the biomass, their number, their quantity, their amount increases to a large extent. And finally, thus these Escherichia coli, which have a recombinant DNA, they grow in large number in this, and then they undergo transcription because our recombinant DNA is there in them. The Escherichia coli, which is growing here in the nutrient medium, it has a recombinant DNA. So this recombinant DNA also grows. I mean, when they multiply, when they increase in number, so many recombinant DNAs will also increase in number. And those recombinant DNA which are there in the Escherichia coli, they undergo transcription and then translation, and they form the protein. Whichever for whichever protein the desired gene is present in them, they will form that protein. Here we took the example of insulin, that insulin is formed. So insulin is formed in this case. So what happens now when their biomass increases in the bioreactors? What do we have now? Now we have nutrient medium, we have bacteria which are transformants, and the product form. Product form means a protein which is formed by the desired gene. Now we have these three things, whether we have done this in this bioreactor, or we have done this in the sparge stir type bioreactor, or we have done in simple stir type bioreactor which has a motor. This doesn't have a motor. Okay, so whether, and see, this is the rotator here again. It keeps on rotating and mixing up the nutrient medium with the inoculum, and also with the air which rushes in, the bubbles which are formed, all gets mixed. Now, so we want our product now. We want to separate our product. How that is done? So after we obtain this, what is done next is, in both the bioreactors, whether we use sparge stir type or simple stir type, now we have the nutrient medium, which is there, the nutrient medium, plus we have the bacteria, which are transformants. Why transformants? Because they have a desired gene in them, the recombinant DNA in them, so we call them transformants. And along with this, we have the product which is formed. We have the product which is formed, which is mostly a protein, which is formed. That product is there. Now we want, see, now we don't want this nutrient medium. The nutrient medium was there for the bacteria, because the nutrient medium has all the type of vitamins, salts, minerals, which fed the bacteria while it was growing. So this is not required now. The bacteria has done its task. That means it has produced the product. Now we do not want this. We do not want this. We do not want this. We are just concerned with a product and we want a product. Now, how this product is taken now from this um, solution, how this is taken out from the bioreactor. So there is an outlet from where this is taken out. And finally, this process by which the product is separated is given a term, and that term is downstream processing. Downstream processing. So downstream processing is separation of product from nutrient medium and bacteria. So this is what about the downstream processing. Then downstream processing is done to separate our product which is formed in the bioreactors from the nutrient medium and bacteria. Now the first step in this is isolation. So first thing which is done is isolation. The product which is formed is isolated by various techniques. So isolation, first isolation is done, and after isolation the product which is obtained, this is obtaining the product, this is obtaining the product, and after the product is obtained, the product is in the second step, the product is purified. So we write it purification of the product, purification. So it is purified, product is purified. We get purified product as a result, which is isolated from, which is separated from all the impurities, all the minerals, all the salts, whatever is present. We do not want anything, no bacteria, no nutrient medium. So it is purified. So the next step is purification. After purification, we obtained a purified product, which is tested. So the third step is testing. So now the product, suppose insulin which we have received, how is it working? Whether it is suitable for human beings or not, or whether it is suitable for mammals or not. So this is done. The testing is done at various levels. At various levels. Like these days, the testing of corona vaccine is going on at various levels. First of all, in the animals, then few people, then the number of people increase, and then again at the third level, and they in between, some days are kept so that they come to know whether the vaccine is showing some effect or not. So same way, the product obtained is tested first on animals, then the experiment is kept, means they are injected or they are given various doses of this particular product, and they are observed for few months, few days, or few weeks. And after that, after the results are positive in case of animals, the product is tested on some, on few people. That is the first level which is tested among the people, among the human beings. So that first level of test on the human being is done. Again, various doses of the particular product which is formed is given to the human being, either orally or by injection, and then their effect is observed for few weeks or few months. And then the next level, that means the number of people are increased, the number of doses are increased, and then the third level. And finally, when the results come positive, then only we say that fine, this vaccine or this particular product which is formed as a result of biotechnology is fit for human consumption. So the testing at various level takes place. After the results are positive, after the results are favorable, after the results are favorable or positive, favorable, then what happens? The packaging is done, packaging of that product, and then and commercialization. Commercialization, that means it is sold in the market by different names, okay, by different trademarks, by different companies. This is the name, it is labeled and then sold in the market. So all this comes under the downstream processing. when the product which is made as a result of biotechnological process in the bioreactors, it is isolated, purified, tested, packaged, and then sold in the market, which can be used by the people, which is fit for human consumption. Now here we took example of insulin. So what is sold in the market? Insulin is sold in the market under different brand names, different companies, they take it and then finally they sell it in the market. The people buy it, the